Hello everyone, my name is Uyen. I'm doing my PhD at Trobe University in Australia. Today I'm going to present the project I have been working for the past few years, Insight into Opium Poppy Genetic Diversity from Genotyping by Sequencing Analysis. This work was conducted within a collaborative research project between La Trobe University and the industry partner Papaver. Opium poppy, which scientific name is Papaver somniferum, is an annual plant belonging to the Papaver genus. It is well known as one of the oldest medicinal plants and the only species can naturally and efficiently produce morphine in the world. Besides that, its long history of cultivation has resulted in its global distribution too. However, because nowadays the movement of poppy germplasm between countries is strictly regulated under the international law, our knowledge of poppy's global genetic diversity is limited. Therefore, our main objective research was to characterize the genetic diversity of opium poppy, obtained from a global germplasm collection using genotyping by sequencing. A detailed understanding of genetic diversity of opium poppy will be helpful for poppy genetic resource management and genetic improvement. Let me briefly introduce you about genotyping by sequencing GPS technique that we apply in this project. GPS is a method to detect the single nucleotide polymorphism or SNP as genetic markers based on the reduced representation library. The GPS method includes four main steps. First is to make a reduced representation library by digesting each DNA sample with one or two resistance enzymes. This is a critical step because these resistance enzymes will be specific for each species based on the characteristics of each genome. In the second step, the digested fragments in certain size, usually from 150 to 450 base pair, will be ligated with the barcode and then go to the sequencing step. The last step is to proceed sample sequencing with index barcode adapter to produce the DNA sequence for each of our sample. With this reduced representation library, GPS shows a helpful method for genetic diversity analysis for a huge number of samples, especially in species with a big genome such as 2.87 gigabytes in opium poppy, which will cost a lot of money for doing whole genome sequencing instead. To apply the GPS technique in our study on genetic diversity of opium poppy, we start with a global poppy collection including 90 accessions from 30 countries of Europe, Africa, America, and Asia with a high diverse morphology such as flower pattern, capsule shape, as well as seed color, in which there are 87 Papaver somniferum accessions and 3 Papaver sativum accessions. Sativum is believed that it is the wild ancestor of somniferum, and it is also the only species together with Papaver somniferum shows the ability to produce morphine in nature. To generate a SNP dataset sufficient for genetic diversity analysis, we first optimize a GPS protocol for opium poppy, especially in the first step to choose the resistance enzyme for GPS library preparation. Among 8 combinations of 2 resistance enzymes include 1 rare and 1 common cutter. EcoRI combining with NLA3 was selected as the optimal resistance enzyme combination based on 2 criteria: the absence of visible repeat regions within the site selection area and level of amplification. Moreover, SNP datasets which were cloned from this GPS library show evenly distributed across within and between 11 poppy chromosomes. From these results, we conclude that our optimized protocol with EcoRI and NLA3 resistance enzyme could allow researchers to rapidly apply GPS to unlimited number of poppy accessions in the future study. To analyze the population structure of 90 accessions, we use the SNP dataset to go to BIC analysis. According to the lowest BIC value, 5 clusters or subpopulations were identified from the 90 study Papaver accession. Then to understand the genetic relationship between the 5 subpopulations, we did the DABC plot. From this plot, we can see that subpopulation 1 were comprised of on 3 Ascetium accession, while on others were Soniferum accession. The wide separation illustrates the extensive genetic difference between Soniferum and Setigerum. To continue investigating population structure in greater detail and detected admixture within 90 puppy accessions, we applied the amateur model in structural software to clustering the accession using about 49,000 unlinked SNPs. The most proper K value detected from 4 to 8. Using bar plot for each optimal K value with the accession sorted following the DABC result, we can see that the pattern of subpopulation assignment did not change significantly across the different K value and were consistent with the 5 subpopulations determined by DABC. At K equal 5, subpopulation 1 were composed of on 3 Papaver Setigerum accession, which are genetically distinct from on other accessions in purple color. Interestingly, subpopulation 2 were a less genetically diverse group which contained 12 accessions of broad geographic origin, extending from North Africa to East Asia. This result could indicate the germplasm exchange in the past. Subpopulation 3 contained the 4 accessions from North Korea with moderate level of admixture from subpopulation 2 and 5. Subpopulation 4 were composed of 21 diverse accessions with a high level of admixture from subpopulation 2, 3, and 5. Subpopulation 5 contained 50 accessions with low level of admixture. Although the 5 subpopulations show no strong association with the geographic origin at regional levels, by combining the hierarchical clustering and population structure analysis with country information, we recognize that some accessions often from the same country were grouped together. For example, here are accessions from North Korea, Japan, Morocco, and Mongolia. These results could be the reflection of the highly restricted movement of puppy germplasm between different countries or regions and the controlled circumstances under which puppies are cultivated. To explore the phylogenetic relationship between study accessions, we construct a neighbor nest using unlinked SNP. Distinct genetic groups were identified and mostly reflecting the subpopulation identified by the population structure analysis. On 3 Setigerum accessions in group 1 and on other Soniferum groups were clearly separated. The accession of subpopulation 4 differentiated into 2 groups, group 3 and group 4, consistent with the structure analysis, which indicate the highest amount of admixture in this subpopulation. Moreover, we found that variation in thousand seed weight and the number of branches per plant were broadly consistent with the subpopulation and phylogenetic group were identified. The Setigerum accession in subpopulation 1 and accession from subpopulation 3 and 4 produced lighter seed and were highly branching. Contrastingly, accessions in subpopulation 5 produced heavier seed and were less branching. The association between branch number and thousand seed weight was significant negative correlation. Although this finding needs more further verification, the data can be a useful input for studying the domestication history of opium puppy, as well as for the genetic improvement for this variable group, especially under the impact of climate change. On the oncolytic profiling, we observe a considerable variation among the accessions in oncolytic content and composition. Although morphine were dominant oncolytic in most of accessions, we also identify accessions with codeine level of up to 54% of total oncolytic We tested the relationship between genetic and chemical diversity by applying principal component analysis. The accession of subpopulations 4 and 5 had more similar alkaloid composition. The centigram accession was clearly separated from others, while accession of subpopulation 2 in green color had the most diverse and distinct alkaloid composition among the somniferum accession. With all of these results, the conclusion for our study is EcoRI and NLA3 were an optimal combination for opium poppy GPS library preparation. Phylogenetic groups mostly reflect the 5 genetically distinct subpopulations identified by population structure analysis. Most accessions often from the same country were grouped together within subpopulation. Agronomic-related traits such as number of branches or thousand seed weight were broadly consistent with subpopulation and phylogenetic groups. To get more detail on our study, you can get our paper from scientific report with this link. I would like to give a special thanks to my supervisor team, Mulu, Papna, and Matt, and I'm also appreciate on help from my colleagues in Lucy Lab, as well as Christopher and Arthur from Palapagma. Thanks La Trobe University for my PhD scholarship, and ARC Research Hub for Medicinal Agriculture and La Trobe Institute for Agriculture and Food for funding our research. Thanks for your listening, and I'm delighted to get your question.